Morphogenesis is the biological process that shapes tissues and organs during embryonic development. It has historically been understood in terms of biochemical gradients, like, for example, morphogens, and genetic signaling cascades. However, in recent decades, a parallel layer of bioelectrical communication has emerged as a fundamental contributor to pattern formation, tissue organization, as well as regenerative processes. Developmental bioelectricity refers to the spatial and temporal patterns of voltage gradients created across cell membranes and tissues due to flows of ions, where these electrical states can act as instructive signals influencing the cell behavior and gene expression beyond local biochemical interaction. And today we're going to cover a study that expands on this framework by illuminating the role of electrical field itself, not just a localized membrane potential, as a global coordinator of patterning information during this morphogenesis. For much of modern biology, the formation of shape in living systems has been explained through genes, chemical gradients, and mechanical forces. Morphogenes diffuse, and cells push and pull on one another to sculpt tissues and organs. Yet, this picture has always been incomplete for us, where cells are not only chemical entities but electrical ones, maintaining voltage differences across their membranes, for example, and exchanging ions with their neighboring cells, for example. Now, over the past 2 decades, the developmental bioelectricity has revealed that these voltage patterns are not incidental side effects of metabolism, for example, but they are meaningful signals that guide growth, regeneration, as well as anatomical patterning in the cells. This paper that we're going to discuss today then has take this idea as a significant step further, in that rather than focusing solely on local membrane potential and direct to cell electrical coupling, the authors here argue that the electrical field generated by tissues act as a global informational layer in morphogenesis. And in this view, the biological form is not only coordinated by local bioelectrical circuit, but also by tissue scale electric field that integrates and compresses, as well as feeds information back into the living organism and into the living system. And this reframing has deep implication in that it suggests that biological systems may use physical field as carriers of information in much the same way that engineered systems, for example, use electromagnetic fields to coordinate activity across space. At the center of this study is the concept of bioelectric pre-patterning, so to speak, where long before visible anatomy emerges, tissues exhibit already spatial patterns of membrane voltage that predict future structures such as heads or faces or limbs or organs, for example. These voltage maps act as kind of a blueprint that cells later on translate into physical form. And traditionally, such patterns have been modeled as arising from networks of an ion channels or pumps or gap junctions, for example. While these mechanisms remain essential, still, this particular paper show that they are not actually the whole story. When many electrically activated cells coexist in a tissue, for example, they collectively generate an electrostatic field. This field is not confined to cell membranes, but extends across the tissue, linking distant regions through physical space within the living organism. And the authors here propose that this field plays an active role in shaping bioelectrical patterns by acting as a coarse-grained projections on the system's internal state. In other words, the field summarizes the collective electrical activity of many cells and then feeds it back into the same cells, influencing their membranes, potentials, and interactions within the living organism. And to formalize this idea, this paper draws on synergetics, a framework developed to explain how coherent pattern emerges in complex living systems composed of many interacting parts. In, for example, synergetic systems, global order parameters arise that constrain and coordinate local dynamics. In this context, then, of morphogenesis, the electrical field functions as such an order parameter where it imposes global structure on local bioelectrical networks, enhancing pattern stability and increasing the richness of possible configurations within the living organism. One of the key findings here is that pattern complexity is maximized when the electric field is both highly causal as well as highly compressed. Causality here refers to how strongly the field influences the behavior of the individual cell, while compression refers to how efficiently the field represents the collective state of the system. When the field meaningfully shapes cellular behavior while summarizing information at a global scale, the resulting bioelectrical patterns then become more complex and expressive rather than those generated by local interactions alone. This result points to an important conceptual shift. Biological patterning is often described as bottom up, with local rules giving rise to global form. The field mediated model here then introduces a complementary top down influence where the tissue scale electrical field constrains and guides local cellular decisions, creating a continuous loop between local activity and global structure. Information then here flows not only from cells to tissues, but also from tissues back to cells. Researchers here then explore how this field mediated system can be deliberately influenced using computational models. For example, they show that transient external electrical fields apply at specific boundary regions of an tissue can steer the information and formation of bioelectrical patterns in the interior of these cells. These external signals do not then need to be permanent or strong. Even brief penetration can bias the system towards particular configuration, after which the tissue's own dynamic carry the pattern forward, for example. And this finding has important implication in that it suggests that morphogenetic outcomes may be shaped without directly modifying genes or biochemistry, simply by nudging the electric field that coordinates the cellular activity. Such intervention act not by micromanaging cells, but by influencing the environmental that the information is within in which they operate. A very interesting aspect of this study was the identification of two distinct pattern coding strategies that emerged depending on how sensitive the cells were to the electrical field. In one regime, where they called it mosaic coding, the pattern information was encoded directly in local membrane of the cell's voltages, where each region carried explicit instruction as to what it would become. In the other regime, called stigmergic coding, the cells responded more strongly to the field itself, the electrical field, and pattern information was distributed indirectly through modification of the shared electrical environments. Stigmergy is a concept borrowed from studies of collective behavior, such as termite or mound construction or ant forging, where agents coordinate not through direct communication but by altering a shared medium. In the bioelectrical context, the electrical field became that shared medium per se, where cells left traces in the field that influence the behavior of other cells, allowing complex patterns to emerge without a centralized control. And interestingly, this model reproduced key qualitative feature of the bioelectrical pre-patterning observed, for example, in frogs. This alignment with experimental observations suggested that field-mediated coordination is not merely a theoretical possibility, but may reflect how real biological systems actually organize large-scale anatomical structure. Beyond developmental biology, the ideas in this paper resonate with broader questions about information in living systems. The electrical field functions as a non local carrier of information, integrating signals across space and time. It is not tied to a single molecule or pathway, but actually exists as a physical structure that both reflects and constrains biological activity. This challenges the notion that biological information must always be localized or discrete or molecular in nature. Instead, this work points to a layered view of biological information where genetic networks operate at one level and biochemical gradients at another level, and also bioelectrical fields are at yet another level. These layers instead continuously allow living systems to coordinate behavior across scales in ways that are robust, flexible, and adaptive. Thinking into the future, the practical implications of such work and such perspective is substantial. For example, in regenerative medicine, manipulating electrical fields could potentially provide a way to guide tissue repair and regeneration without invasive procedures or genetic modification. As well as in bioengineering, for example, understanding field mediated pattern could potentially enable the design of synthetic tissues that self organize into desired forms. More broadly, this research invites a reconsideration of how physical fields participate in biological computation as well as decision making in living systems. This type of research also maps well onto the concept of symbiotic intelligence, especially in how intelligence and coordination emerge from interactions across multiple scales rather than residing in one single locus. In, for example, this framework, biological systems are not merely signal generators, but active participants in a distributed information ecology, where meaning arises through interpretation, feedback, as well as integration. This paper's treatment of the electrical field as a global causal medium aligns directly with the symbiotic intelligence in that this field is not just carrying signals but shaping how signals are interpreted by, for example, cells, effectively acting as an intermediate intelligence layer between local biological units and large scale structure, where this mirrors the argument that intelligence can emerge from continuous coupling between local biological processes as well as higher order organization substrates rather than from just discrete computational units alone. And the stigmergic regime described in this paper, for example, so strongly also echoes the emphasis in indirect coordination through shared informational environments. In symbiotic intelligence, for example, human biological signals such as brain waves or heart rhythm, etc., are meant to be the bioelectrical patterns to be read an isolated data point, but also as part of a living or adaptive loop between the humans and the machines. And the same way the bioelectrical field model, cells do not simply transmit instruction to one another, but modify a shared electrical landscape that in turn guides collective behavior of the entity. And this suggests a deep biological precedent for the idea that future intelligence system will operate by co-shaping an information field with humans rather than by issuing commands or predictions in isolation within, for example, humanoids, where the electrical field becomes a biological example of symbiotic interface, one where intelligence is not owned by any single agent, but arises from sustained interactions between the components across space, time, and scale between the biological entities and the artificial entities. Living systems such as you and I already use the electrical field level information integration to coordinate complex outcomes and complex information processing long before the digital computation or artificial intelligence extended. In this sense, the symbiotic intelligence here then proposes that artificial systems can participate in similar loops as in living system by coupling to human bioelectrical signals in real time. In that sense, symbiotic intelligence is not just an abstraction layered onto biology, but it's a continuation of the same informational logic that morphogenesis, for example, already uses in living organisms. And the implications here is extremely powerful in that if biological form is shaped by shared electrical field of meaning, then future human machine systems may achieve genuine integration not by mimicking cognition, but by learning how to inhabit, interpret, and co-regulate the same informational electrical field that the living systems such as you and I have already and always relied upon. And that's all I had for today. Thanks for tuning in and thinking with me, and I'll see you guys next week. Bye.